This talk, I was running late because I was trying to put together my slides. It's a new way of giving this talk. I'm kind of giving my scientific history and my scientific path and all the different decisions to get to where I am today. So my title slide, hopefully you can see that it's a movie theater, and that's relevant to my scientific path. I grew up in, I was born in Illinois, in the Midwest. Oh, guess. Thanks. So I was born in Illinois, but I grew up in Paris, but that's Paris, California. So most people, when I say Paris, I don't know who's heard of it, but it does exist. There, when I was growing up in elementary and middle school, my memories of science actually were through the television shows Square One TV and Mr. Wizard's World. So some of you might be too young to remember that, but I don't know how many people ever watched that show. Wow. I'm actually shocked that they're amazing. There's probably YouTube videos for some of them, but Square One TV was actually focused more on kind of math. It was kind of like Saturday Night Live, so little skits, but they would teach math concepts. And then Mr. Wizard's World, Mr. Wizard would do scientific experiments with kids. I loved these shows. So those were kind of my early memories of science and math. But then when I started high school, this movie came out, Gattaca. People have probably watched Gattaca. Yes. If you haven't watched it, I highly recommend it. I think it's very relevant to kind of think about these concepts of how genomics can affect society. But basically, I watched this movie, and what it's about is actually it's more of a dark outtake on genomics. So what are some of the bad things that can happen if given genomic information? But I won't spoil the movie if you haven't seen it. But the basic thing that I came out from watching this movie is, wow, it's really cool that someday there could be a future where you get some genome sequence at birth, and then the nurse puts that genome sequence or the DNA from birth, and you can sequence the DNA right there, and then you get this printout of your probability of having being obese, the probability of having a heart disorder, and your life expectancy. So I came away more with, oh wow, that's really cool that you could take genomic information and just predict phenotype. So I decided that I was interested in genetics. Didn't really know anything about genomics at the time, just from that movie, but then I decided to go to UC San Diego, so stay kind of close by to home. Yeah, go Tritons. And so when I started, I was like, okay, well, I want to do genetics, so that's kind of like biology, so I guess I'll do pre-med. So I started as a pre-med, and then over time, I kind of realized, well, I still like math, so I was mentioning that Square One TV show. I was always kind of interested in math, so I thought about changing to a math major. And then the beginning of my sophomore year, this new major was created called Bioinformatics, and I was like, what's bioinformatics? I don't know. And I actually remember going to the undergrad advising office where they had the sheets of different majors and like what the classes you took and the description of the major. And I remember reading it and realizing, oh, like you can analyze DNA and genomes, and so it just seemed like a really good fit. So what I guess my view of what bioinformatics and computational biology are is, again, this is from my interest. You can take from some DNA sample, you get some genome sequence out of it, you further analyze that in a computer, and then potentially, get some phenotypic prediction of based on that genomic sequence. And all along this path, each of these steps require bioinformatics or computational biology. It also requires intuition about biology to make sense of the information coming out from those analyses. So I thought, okay, that bioinformatics seems pretty cool. It fits all my interests. And but I had never taken a computer science class. I had never programmed before. I liked math, so I figured, it sounds like it might be logic, quantitative. And I took my first computer science class, and I really loved it. It was kind of like playing a video game to me. You're just kind of thinking about it, the way you have to think about a problem when you write a program. So it just seemed like a really good fit, and I decided to be a bioinformatics major. So it was cool. It was a mixture of biology and computer science. I took somewhat lab stuff. And as I was going through coursework, and I kind of decided, well, I don't know if I want to be a pre-med anymore. And because I was really interested in just kind of the understanding how cells work and understanding how, or trying to interpret genomes. And so then I was considering grad school. I didn't really know what it was or what you do in grad school. And I remember going to the grad school fair and reading a brochure about different grad schools. And I was like, oh, it's free and they pay you. So I figured, I know it's not a lot, but I mean, it's very few paths where after college you actually don't necessarily go into more debt. So then I kind of started to explore what grad school was like. And so I found out about these undergraduate research programs that I could be involved in, so I could learn more about grad school and what research was. So I applied for the UC Leads program. It's a UC-wide program. And actually, as part of that, I did my summer research here at UC Santa Cruz with Todd Lowe. So he's actually chair of the department now, and so now I'm a faculty in that department, which is really awesome. It's like kind of crazy. But that research experience was really transformative for me, where that was the first time I'd actually taken the stuff I'd learned from classes and applied it to a research problem. And that was also my first experience into the RNA world, and so I really enjoyed that. And then I also did summer research in LA. So that's to say that, and I guess for the students that are here, if this is your first opportunity, I highly recommend looking into other programs. It's really nice to get a diverse sense of different labs and different institutes as you're gaining experience in research. Okay, so then got some research experience and decided to go to graduate school and stay in California. And I worked with Stephen Brenner, and my thesis title was Alternative Splicing Regulation in Drosophila melanogaster. And I'll go through what that is a bit. So back in 2000 or so, groups, including the Hausler Lab here at UC Santa Cruz, sequenced the human genome. And after looking at the number of genes that were found in the human genome, these groups compared them to the number of genes in other organisms. And it was really sad for people to see that the number of genes in the human genome were not that far away from the number of genes in other species such as worm and fly. And so it was puzzling. I mean, people expected, humans are more complex organisms people, you could, I guess, population geneticists or evolutionary biologists would argue that. So, but I guess humans are thought to be more complex, particular with neuronal development. But anyway, so the number of genes were about the same, and so that was kind of puzzling. And so one layer of information that's missing when you just look at the number of genes is actually the number of products that can be processed from those genes. And so it's thought that that the complexity that's observed in humans is partly attributed by the number of transcripts that come from those genes. So what do I mean by that? So here I'm diagramming a gene locus that And genes are organized into exons and introns. So when a gene is initially transcribed, you get a pre-mRNA. And then those intronic sequences are then removed by a process called splicing. And then you end up getting multiple different types of mRNA isoforms from that gene. So from this one pre-mRNA, you can decide different exons to choose, and that leads to different mRNA isoforms, which then leads to different proteins, and each of those isoforms can have a different function. So in my graduate thesis, one of the questions I had was, how does the cell know to splice, say, in a lung tissue to keep all 5 exons, and then in skeletal muscle, how does the cell know to choose to skip this blue exon and connect this first exon to that third exon directly? And so those decisions are done by RNA splicing regulation. So this is, again, kind of a schematic of a pre-mRNA with here just showing 3 exons and then intervening introns. And along that pre-mRNA, there are sequences that help to regulate that process. Some of the sequences are described as enhancers or silencers. And what these sequences are, they tend to, or they act as binding sites for proteins that help to recruit the splicing machinery to certain exons to tell the splicing machinery to, say, keep an exon. So the arrows are just kind of showing activation. And then there are other proteins that can act as repressors, where they bind nearby and then act to repress the splicing of those exons. And so for my graduate thesis, I was trying to study what these sequences are and what were the proteins that were involved in this regulation, but I was doing this in Drosophila. So a few ways, so this is actually kind of a summary of my thesis. So in one study, we did a computational prediction of those splicing regulatory sequences. So what are those sequences that are potentially in the pre-mRNA that are helping to control splicing? We were looking at RNA sequencing data to actually analyze and identify alternative splicing changes, and I'll talk about that a little more later. We also sought by knockdown experiments to try to identify what are the proteins that control splicing by knocking them down and looking for differences in splicing, and then actually linking each of these RNA binding proteins to the targets, to their targets. And then finally, we were interested in understanding how these splicing changes affected or changed throughout Drosophila development. So we looked at sort of a time course of splicing changes. So then that was the end of my graduate career, and I was trying to decide what to do next. And as I was looking back, this is a real slide from my last thesis committee meeting in graduate school, and I was talking about some of my long-term plans. And I was basically deciding to do a postdoc afterwards, and I was pretty sure I wanted to go to academia, and I figured, even if I did an academic postdoc, I could go to industry, or if I went into industry eventually, I could probably go back to academia. But some of my main interests were understanding how non-coding variation affects phenotypic variation. So when there's alterations in portions that don't code for protein, kind of trying to understand the function of those. And then I was also interested in personalized medicine. So right now that people don't really call it personalized medicine, they usually call it precision medicine now. So with those themes, I decided for my postdoc that's kind of what I wanted to pursue. So I decided to go into eventually go into the field of precision medicine, but in the context of cancer. and because I think cancer is one of those diseases where precision medicine has really been shown to work. And so the idea is that if you have a cancer patient or a selection of cancer patients with different sets of mutations, you can identify certain mutations that a subset of patients have and then know that there's a particular therapy that would work very well with those patients. So then I thought, okay, cancer genomics seems like an interesting field. We can sequence a bunch of patient cancer genomes and try to identify all the mutations that occur in cancer so that we can identify potentially new mutations that could be targetable. And what I'm showing here are, so currently if certain patients and certain tumor types, if they carry an EGFR mutation or a BRAF mutation, there's a particular targeted drug that can match that. And so I became interested in how do we look for these other mutations. And so the way cancer genomics works, and another reason why I was interested in cancer genomics is you have a nice control for these patients in that you have mutations that occur in the tumor tissue DNA, and what I'm showing here is in each of these gray bars is an individual segment of DNA that had been sequenced from that patient tumor. And if you align those DNA sequences to, say, the human genome reference, you can find specific positions that differ in the tumor DNA compared to, say, the genome reference. But what's nice with typically with cancer genome patients is then you also get a matched normal tissue. So you can be really confident about mutations that happen specifically in the tumor because when you sequence the normal DNA, there doesn't appear to be any mutation at that position. So if we do this for hundreds and thousands of patients, so this is from a study in 2014 where they did this on 230 tumors, what you can then find are commonly mutated genes across many patients. And the idea is that if you look then at genes where a large proportion of patients have that mutation, then that can represent a common mechanism of cancer tumorigenesis, and then that can also represent a potential targeted therapy. So I was really interested in going into this field. It was something that I didn't study cancer. My background was in Drosophila RNA Splicing Regulation, but I was really interested in moving into this cancer genomics field. I thought it was really interesting. What I decided to do for my postdoc is to go all the way to Boston on the East Coast. And I also recommend that for people. It's nice to stay, and it was actually really hard for me to leave California for my postdoc. I notice I'm back because I really didn't like the winters, but in the end, I actually miss Boston a lot, but it was a really good experience to get out of my comfort zone and experience this new location, a new field. So it was a really great life experience for me. And I was fortunate to work with a lung cancer genomics expert or general cancer genomics expert, Matthew Meyerson. One reason why lung cancer was interesting to study is because it is one of the leading causes of cancer deaths in the these are statistics for the U.S. And also it's the second most common type of newly diagnosed cancer amongst men and women. So it's a very prevalent form of cancer. And the other reason why it was interesting to learn about lung cancer and lung cancer genomics was currently it's one of the cancer types where there is a targeted therapy that is a standard of care. So So if someone has lung cancer, many lung cancer patients get this gene EGFR sequenced because there's a specific EGFR inhibitors that those patients respond very well to. And actually in initial studies of this inhibitor, they found that patients that had this mutation, if you gave them standard chemotherapy, they actually did more poorly than if you didn't have the mutation. So identifying patients that had this mutation was really important in the treatment decisions. So as part of this group, I was involved in studying lung cancer genomes. And what came out of these studies, again, we sequenced many patients and looked at what were some of the commonly mutated genes. We found that there were many genes that were involved in splicing. Many genes involved with splicing or had an alteration due to splicing. So what this figure is showing is in the columns, each tick mark represents a patient, and each row is a gene, and the colors are different types of mutations in that gene that were identified. And so it seemed that actually splicing seemed to be a common way that cancer genomes were being altered. And that was actually very surprising for the cancer field. At that time, no one had really thought about splicing as a way for cancer cells to be mutated. And so it was pretty much serendipitous that I had this huge splicing regulation background in my graduate work. And so I decided to pursue this area of research to kind of understand more about these splicing alterations in cancer. So one of those recurrent mutations that were found in lung cancers was in this splicing factor called U2AF1. So if you remember, I had the slide where splicing regulation is controlled by these proteins that bind to the RNA, and one of those is U2AF1. And so the U2AF1 protein is thought to help recognize the 3' splice site of introns. And what was striking about these U2AF1 mutations, and now this is a look of a particular position that gets mutated highly frequently across multiple types of cancers. So it was really striking that in all these different cancer types, the same exact amino acid position was found to be mutated. And so that is a common feature, again, if you see the same missense mutation in many different cancer patients, that's a signal that that's a very selected mutation that's advantageous for the cancer cell. With further cancer genome studies, many groups identified that these splicing factor mutations are occurring in many different cancer types and also different types of splicing factor mutations. So I mentioned that U2AF1 protein, but there are many, many other proteins involved in regulating splicing that have been identified as mutated in cancer genomes. And so one of the questions in the field, and this is still pretty much an open question, are how do these mutations really contribute to tumorigenesis? So one idea is when you have a splicing factor mutation, potentially you're altering splicing. So here I'm showing a diagram of alternative splicing where you can decide to include this exon or decide to skip it in the eventual mRNA. So one hypothesis is that these mutations are affecting the splicing of these exons. Another hypothesis is that maybe you're getting intron retention or just not splicing at all. Or maybe you're just getting general gene expression changes. And that somehow the collective action of all these splicing changes or RNA level changes are affecting some specific genes or pathways. So fortunately, with a lot of cancer genome studies, particularly the data that we work with in the Cancer Genome Atlas, there is for each tumor, there's the whole exome sequencing, but there's also mRNA sequencing. So we can actually look in the same tumors that have those mutations and ask, is there something different about the mRNA? So our approach then to look at to ask what are happening in those patients that have these splicing factor mutations, we compare the transcriptome, so it's a collection of all the mRNAs, between wild type and mutant tumors. So you can take the collection of patient samples that are wild type for that splicing factor, and then a collection of patients that have one of those splicing factor mutations, look at the RNA-seq data and compare them and ask, is there any differences in mRNA splicing? So in my graduate work, since I was interested in looking at splicing regulation in Drosophila, I built this tool called JuncBASE that can take RNA-seq data and look for all these kind of categories of splicing and also perform differential splicing analysis. So I decided to use this tool and update it so that it could work for human RNA-seq data. But I guess just a take-home message from the slide is that what JuncBASE can do is look at many different forms of splicing. So how does JuncBASE actually quantify those splicing changes? So what I'm diagramming here is a type of alternative splicing where the black exons represent exons that are always kept in the mRNA, and this white exon is an alternative region, so that piece of exon can be included or skipped in the eventual mRNA. And in red, I'm showing I'm diagramming a sequenced read that would support that the alternative exon is included. So that would be a read that is split between this upstream exon and downstream exon, or any read that falls within the alternative region. So those red reads support the inclusion of this alternative region. And then skipping of that alternative region would be supported by a read that is split in its sequence by aligning upstream to this exon and then partially downstream at that constitutive exon. So what JuncBASE will do is it will find these reads and then calculate an abundance from that. So it's a number of reads over the length of the isoform. And then from that, it calculates a percent spliced in. So for one given alternative splicing event, a percent spliced in value will give you what's the relative amount of this alternative exon relative to the total expression of that region. So from that one PSI value, we can use it to compare the splicing differences between a collection of wild-type tumors to a collection of tumors that have a particular mutation. So I'm showing these are percent spliced in values for patients that were wild-type for this U2AF1 gene, and then this is the PSI values for tumors that had this S34F mutation. And so by comparing just these values using a Wilcoxon test, we can test individual splicing changes to see if they're different between wild type and mutant. And by doing that, we did this across lung adenocarcinomas and acute myeloid leukemias, which is another cancer type. And we found 130 events significantly associated with those mutations in lung cancer, and then about 369 in AML. And the colors here in the bar represent the different classes of alternative splicing events that were identified. And so what you can see is that a majority of the splicing changes we found were either a cassette exon type or alternative 3' splice site. and that was consistent with what we knew about the function of U2AF1. So we knew the normal function of U2AF1 was to recognize 3' splice sites, and given that mutations in that gene preferentially alter cassette exons and alternative 3' splice sites, that all made sense and pointed to the mechanism of how these mutations are acting on the cancer cell. So one thing that was sort of challenging in this analysis was that we identified these hundreds of mutations that were we identified hundreds of splicing events that were altered due to this mutation, but one of the challenges was the not understanding what the functional consequences of these mutations were. So for example, we saw these key genes and certain pathways that were being alternatively spliced, but because we didn't really know how that splicing event changes the function of the gene, we really didn't know how this affects the downstream pathway. Okay, and so in addition to, all of that was a study we published in my postdoc work. And in addition to these splicing factor mutations, such as in U2AF1, another way that we've been looking at splicing factor mutations in cancer genomes is through splicing regulatory sequences. So in the pre-mRNA, you have sequences that also help to guide where to splice, and I had mentioned this earlier. And so we are also looking at mutations that occur in these regions. So one of the classic examples of this is in this gene called MET, which is a known oncogene. And what had been observed previously, and we found more evidence of this, is that there are multiple mutations that occur around this exon 14. Some of them are small deletions or point mutations right at the regulatory sequence. And when patients that have these mutations have skipping of this exon because it can no longer be recognized by the splicing machinery. So normally, without the mutations, you have the presence of exon 14 at the mRNA level, and then at the protein level, that exon contains a binding site for the Sybil protein, which is a ubiquitin ligase. And so when Sybil binds to MET at the protein level, it helps to tag it for degradation. But when you have one of these mutations that then causes skipping of exon 14, you lose that binding site, and so that protein can no longer be tagged for degradation. And so you get activation or overexpression of that protein. And so also in this cancer genome atlas data, we wanted to look for these cases across lung cancer. So we again took the whole exome sequencing information and mRNA sequencing information to identify how common is this kind of alteration across lung cancers. And what we found was that it is actually a fairly common mutation. So what I'm showing here, I showed it in a previous slide, but this is the collection of known oncogene drivers. So the idea is that if a cancer has a mutation in one of these genes, that mutation is actually causing the proliferation and oncogenic processes in the cell. So this is a pie chart of all the oncogenic driver mutations we identified across lung cancers. The blue color are sort of newer oncogene drivers that were identified in those sets of patients, and then green are the sets of patients where we couldn't identify any driver. And so this MET exon 14 case, again, this is caused by, this is a mutation that causes exon skipping in that gene. It's actually fairly common, so about 4% of lung cancers were found to have this alteration. And this is now becoming an interesting target for targeted therapy only within the last few years. So based on the study that we looked very globally at many different lung cancers to show the prevalence of this, it's become sort of more of an interest for pharmaceutical companies to try to develop certain targeted therapies for this. So this is a paper, a recent paper that shows that these two different patients that had this MET exon 14 skipping event respond fairly well to a drug called crizotinib. And so in the red arrows showing the sort of location of the baseline tumor, and then after treatment, you see shrinkage of the tumor. So not only is this splicing event a known oncogenic event, but it appears to be a targetable alteration. And so that was sort of a summary of a lot of the work that I did in my postdoc. And carrying forward, so I started last year here in my own lab, and one of our interests is to continue to mine this huge amount of data that exists for all these cancer genomes, both exome sequencing and RNA sequencing, to identify additional cases of splicing alterations. And so we're interested in looking for additional cases of splicing factor alterations and how that affects RNA downstream. And then also looking at a collection of splicing regulatory sequences and how, and if they're mutated, how that affects splicing downstream. So one of the challenges that actually came out from doing all these studies is that we actually don't understand the function of splicing. So in order to understand how, if we see these splicing changes in cancers, it's actually hard to know what that's doing to the gene function. So what we've now started on is to think about how do you take from a collection of these hundreds of splicing changes observed in say, lung cancers, and sort them out to say, well, this splicing change doesn't appear to do anything to the gene, or there's some subset of exons that are actually driving the cancer phenotype. And so what we've started to design is a high-throughput screen to identify oncogenic skipped exons. And we're using the CRISPR-Cas9 system to target these exons. So the idea is you have all the collection of exons that we're interested in testing to see if they're oncogenic. And we're designing cut sites using the CRISPR-Cas9 system. I'll go over that in a little bit, to target right at the splice site to cause that exon to no longer be recognized, and then it would cause skipping. So the work of Max Marin, who's undergrad in the lab, he's now reanalyzed the lung cancer data set and has identified about 500 putative exon skipping events that might lead to the function of an oncogenic splicing change. So essentially, we're looking for more cases like that MET exon 14. So how we're identifying these is by looking for exon skipping changes that are associated with splicing factor mutation. So we're taking all those U2AF1 exons that were identified. He's also reanalyzed the data to look at splice site mutations, or the cis-acting mutations, and then looking at the RNA-seq to see that you also see you see skipping at the RNA level. And then the other way that we're finding these oncogenic skipped exons is by looking for exons associated with oncogene driver negative tumors, and I'll go through that. But the take-home message is that we're identifying these potentially oncogenic skipped exons because there's some genetic mutation that's occurring, and we also have the RNA-seq data to actually see that there's a skipping of that exon from the actual tumors. So the other category that we're so that last category of exons that we're selecting from are changes that we're observing that are occurring in the set of patients that had no known driver alteration. So the idea is that all these patients that are represented in the red and blue part of this pie chart has some genetic mutation where we think we understand what is driving that cancer, but there's a selection of about 24% of patients where there's no recognizable mutation. So we're digging into this set of patients and asking, are there any observed skipping of exons in those sets of patients, which may explain or may be kind of missing oncogenic drivers that haven't been found before? Yeah.
So does that include loss of tumor suppressors as well in that category, or does that fall?
Yeah, so we're so this only represents oncogenic activation. So we are preferentially selecting for exons that would not be loss of function. So for example, the MET exon 14 case, it's an exon that's a multiple of 3, so it wouldn't cause loss of function or a predicted frame shift. So we're kind of enriching for more of these cases. Mm-hmm. Okay, so how are we targeting these exons? So we're using the CRISPR-Cas9 system. And so how this works is if you have a target sequence in your genomic DNA, you can design a CRISPR guide RNA sequence that is complementary to that sequence that you want to mutate. And then that guide RNA associates with this enzyme called Cas9. And what Cas9 will do, it will cut the genomic DNA at a specific position relative to that guide RNA that you've designed. So and this is a huge tool, a powerful tool that many labs now are using because it's really powerful to be able to cut specifically at one site. So the idea of our screen is that for a given candidate, so we actually do have an interesting candidate in this gene called ERB2, which is a known targetable gene. And so what we can do with CRISPR is cut specifically at a splice site near that exon. That mutation, so that cut site would then cause DNA damage at the site, and so we're hoping that when the cell repairs that DNA damage, it would introduce mutations. And so that's how we would get a mutation right near a splice site. Then after that gene is transcribed, then that mutation would then occur in the pre-mRNA, and that exon will no longer be recognized by the spliceosome machinery, and then it would get skipped in the eventual final mRNA. And so we do have preliminary data that this actually works, where you can cut at a specific splice site and then cause RNA skipping. Okay, so yeah.
What cells are you in when you do this, or like in a culture cell or a cancer cell? Or?
Yeah, so so conceptually we can do this in any cell, but we're starting with lung cancer, so we're using primary lung lines that are normal, that normally aren't transformed. And so the idea is you introduce the skipping of an oncogenic splicing event that would transform the cells to be cancerous. So the idea, and this, so I'm in my last few slides. So the concept, so this is a figure taken from a recent study that I was involved in, where we did this not with guide RNAs, but with other types of genetic variants. But conceptually, what we're doing is taking this pool of guide RNAs. So again, we have hundreds and hundreds of exons that we're interested in targeting. And so we're generating a whole pool of these guide RNAs. And then introducing in pools, sorry, what I'm missing here, sorry, is you infect a pool of, you infect cells that are primary lung cells, and then take those cells and then inject them into mice. This is a mycenograft assay. And so after a time, there will be tumors that are formed, and you can sequence the tumors to identify the selection of guide RNAs that are now enriched in that tumor population. And then you've also sequenced the pre-injection pool, or the pool of cells that you originally infected. And then you can ask, and all these colors represented, say, an individual guide RNA, and ask, is there a collection of guide RNAs in the tumors that are present that are enriched relative to the pre-injection? So this is a direct assay to ask in a high throughput way if we, or any of these exons, if you cause skipping, would cause tumor formation, because those guide RNAs would be enriched in the final tumors. So, so really the goal of that screen is to ask, are there potentially new targetable mutations, because these are mutations that would cause an oncogenic form of a gene, and those typically are nice targets to use for targeted therapy. So I'd like to thank primarily the work that I described about the screen is primarily work from Max, Cameron, and Eva in the lab, but I'd also like to thank the rest of my lab. It's been really great to work with really great people. For the screen help, we're working with Susan Carpenter, Mike McManus. I'd like to thank also my old lab that's helped in initially designing the screen or the methods to do the screen. And also the work in my previous lab was involved in actually identifying these candidate exons. Jesse Boehm's group is collaborating with us for those mouse xenograft assays. And I'd like to thank the TCGA group for a lot of good feedback about analyses, and also the other labs in the Genomics Institute for their help. And Damon Runyon for funding my postdoc and part of my startup as well. Thanks for your attention. So there's time for questions if you have any.
You said at the very end my startup as well?
Yes.
Can you say something about that?
Oh, yeah. So Damon Runyon has a called a Dale F. Frey Award, where they kind of at the end of your postdoc, they within the fellows, they give an additional award of $100K. And so I was able to take that to my own lab and use that as supplement for my startup. It's a great foundation. So if you're a postdoc doing, it doesn't actually have to be like directly cancer research, just research that will eventually lead to an understanding of cancer, then I highly recommend. It's a great foundation to apply to. Yeah.
If you have CRISPR and S34 mutation in your assay,
yes.
do you come up with that?
Great question. We have not done that. So what I mentioned, the screen is not doing introducing the mutation. It's hard to do that in a high throughput way. But we have not done the CRISPR in of the U2F1 mutation. I know people who have. It's difficult to work with. Actually, some of the phenotypes that people are seeing is sort of slow growth when you introduce the mutation. But actually, there's a recent paper that showed that if you co-express the mutation with MYC, then you get a selective advantage. And I don't remember if they used CRISPR. I think that might have been overexpression. Yeah. So it's still kind of a mystery. I think there's a lot of probably additional mutations that need to co-occur to actually see the selective advantage for that mutation. Yeah.
Do you think lung cancer will be cured in your lifetime?
I do think that cancer hopefully will be a chronic disease in our lifetime. So not necessarily cured, but cancer cells evolve once you treat with some therapeutic. There's some subset of cells that gain some mutation, and there's now a new clone that arises with new mutations that you then have to hit. And so I think we are getting there in terms of finding potential drugs or therapies to help target different types of cancer cells. But I don't know if it's hard to tell if there's actually a cure. I think it's going to be continual treatment by different options. Unless you can stop evolution. I don't know if there's, if there is, I mean, I guess that's the one way is if you could stop evolution, maybe.
So we can all start smoking again?
I wouldn't recommend smoking. Actually, yeah, no. One of the first projects I worked on was looking at lung cancer genomes and seeing the rate of mutation in smokers versus non-smokers, and so you definitely are rolling the dice more when you're smoking. You're just making the probability that you're going to hit one of these driver genes, more likely. So I wouldn't recommend smoking. Yeah.
I'm wondering because big data like this, you chose a cancer that you said had a lot of occurrences of cancer. But I'm wondering how willing cancer patients are maybe for other cancers to give their data to research like this, maybe less prevalent cancers or even lung cancer since you've been working on it.
Yeah, my impression and talking to people is that particularly patients with cancer that maybe they have a mutation that's completely not known what's driving their cancer, they're actually very willing to try whatever options are available to them to try to figure out what's going on. So they are willing to at least do genome sequencing. I think, in addition, I think a lot of patients are willing to help other patients that have the disease because they know how much it impacts their lives. So my impression is that there are a lot of cancer patients that are willing to make their data available for research so we can make more sense of the biology there.
So is there other research for other cancers like this?
Oh, definitely, yeah. And I really, I focus on lung cancer because I kind of learned more about the biology of how lung cancer works, and I was able to learn about different experimental tools to work in that system. But definitely, like long term, we're interested in moving into other cancer types as well. And there are other groups that are doing similar approaches to kind of dig into what are the frequently mutated genes in these other cancer types, and then how do we take that information to treat patients. All right, great. If you have other questions, I'm around, so thanks.